So what I would like to share with my presentation is actually our experience on doing the feedback microscopy of different kinds. And this would be the smooth transition from the session number 1. I actually want now to invert the logic. I want to start with the examples, right? Starting with maybe simpler examples and going to more complex examples to show what kind of the feedback microscopy we need to do. And by that, to also talk how to select the tools and what will be the demands for the tools, right? And whether it worth it to go just for the fully commercial application or when it will be worth to try to connect different types of tools. As we already started this discussion, right? So I would be also really happy to have the discussion ongoing. Please don't hesitate if you have any questions at any time, just to ask. All right, just to begin with, so what we think of the smart microscopy, or we used to use the term adaptive feedback microscopy, is pretty much fits to what Raphael was talking before. It's actually allowing to control the microscope workflow from being defined to be decision driven. And this is what people do anyway, right? This is what people do manually over the years. So when you're, for example, on a confocal microscope, right, and you want to get fast, high resolution picture, right? If you're on the point scanning confocal, what you need to do, you want to find your phenotypes, right, to go to define these positions and trigger the acquisition in the simplest case, right? So I start with really the example which became a textbook example over the last years, right? If we are interested in the metaphase cells, so we need to identify them, and we just can, for example, grab with a decent resolution just to identify the cells a low resolution image. So I try to maybe get a pointer, maybe it's, yeah. And then we need to spot the cells. And let's imagine that it's also the live sample, right? And we want to track the cells, and we want to do it as fast as possible. So what user would need to do, user would need to acquire this image, to find, to mark these positions, switch to other settings, and then finally to trigger acquisition, which will bring him to that type of example, right? So that's how it's meant to be, right? And this is, at the simplest case, what you would imagine, low resolution and high resolution image. So, and to automate it, I think that's what was already discussed pretty much. So we have this conventional loop where we have autofocus, and this we should not forget, and I will come back to that several times during my topic, because normally you need to control your focus, and that might imply which kind of control from the software part we need. Then we have the image acquisition part, right? And then we trigger online image analysis, being inbuilt or being external, right? Which identifies as the right phenotype. And then we want to start the high content imaging. But what the high content imaging is, it really depends on your application, right? First, we think about the high resolution imaging in 3D, in multicolors, and then we want to do more. And what is more was already mentioned today several times. It can be bleaching experiment, ablation experiment, photoactivation experiment. It can be measuring FCS. It can be measuring FRET. It can be whatever, right? And this is, to our point of view, has to be user driven. And I would basically come to the examples of the feedback microscopy workflows, which we had to deal with, and to actually want to discuss with you how such a decision can be user driven while we still maintain the integrity and capacity of the systems. All right, so just to begin with, I just want to highlight the part of the applications which I will talk in the beginning, and these are type of applications which I would call imaging applications. So first of all, as in the first example, we have targeted imaging, right? We want to have specific objects, and we want to focus on them and maybe acquire them at high resolution. Then another important part of applications where we want to actually track the object over time, right? The object can be moving, and we already had the example today during the first session, and I'm pretty sure that we will hear also more later. We have different experiments. We have FRAP, photoactivation, photoablation, and so on, and we need the respective functionality to be available from both from the hardware and software point of view. And again, in these cases, we not only want to control the coordinates of our stage, right, and imaging modalities, but also want to control the regions which has to be photoactivated, ablated, and so on, depending on our analysis. And last but not least, of course, we have also the correlative imaging, right? So the same type of tools when we need to have when we want to acquire low resolution, right? And instead of imaging high resolution on the same microscope, we might want to automatically identified region imaged not with this modality, we might need to transfer it to another light microscope, for example, super resolution microscope, or also for electron microscopy, and so on and so forth. Okay, so these are all textbooks, and very often we have also like software implementations for these different kinds of applications, right? If you think of it like each application which would need each time one kind of a decision making, right? But now I want to show you actually a few examples of the either completed or the ongoing projects where this was not somehow following the textbook examples and where we needed some kind of extra functionality which we needed to introduce one or another way, right? So let's start with, again, the targeted imaging example. So we have multi-well plate, and here we just display one well where we have Drosophila embryos, right? And what we're interested, simply we need to find these Drosophila embryos, each and single one, or maybe in the beginning, and then we wanted to also classify phenotypes and image them at high resolution. So what one would do, one would say, okay, if you have a protocol, low resolution imaging, high resolution imaging, that's fine. You get a low resolution image, you acquire 5x, maybe tile scan, then you allow either manually or automatically define all the Drosophilas. This can be segmented, and then you switch to the high resolution imaging at 20x, right? And this is your image. And this is a workflow which you want to run over days, or overnight, or over several days, or even longer. So when we started to work with this on this workflow with the real samples which were mounted, which it appeared not to be as easy as it is, because immediately we were, by doing it manually already, we were realizing that there are several improvements which can be done, which will greatly simplify our workflow, right? But not always these improvements are available to do it automatically. So what we realized, sample can be mounted at different heights, right? Because depending how it's mounted, right, maybe it's not all embryos were perfectly flat. So not only you want to select it in XY, you want to select it in Z. If you want to select XY in Z from the beginning, you have to sample in high 3D resolutions, the sample from the beginning. What would be the advantage instead, right? Could we actually separate the decisions? Can we decide on defining XY location at once, and then focus already in this embryo and define the Z location? Another point, of course, if you are on the point scanning confocal microscope, do we need to image always the bounding box, or can we also adapt the rotation of the field of view, for example, and such kind of thing? So at the end, via several iterations of improving this protocol, we ended up, instead of having one feedback step, we ended up to have 3 feedback steps. Okay, 2 of them were essentials. Third of them, it simply came because we were not able at that point control the hardware autofocus together with the feedback microscopy. It was just not fitting together with these tools. So we defined 4 imaging jobs, right? And we actually, each time we had a feedback step. So first we had the XZ scan for the reflection, right? We just identified the cover slip as you would always do. Then using the feedback microscopy, we're defining Z level, and based on that, we were already acquiring the low resolution image, which was in focus with 5x low NA objective. Then this image was acquired and automatically processed. And from this image, we were identifying the embryos, one or multiple embryos, which we want to image. And then before switching to the final scanning, we actually had the step in between where we had actually already with that objective scanning at lower resolution and basically higher range Z-stack, when we were able first to focus on individual embryos to refine its position. And then we could also define the rotation, right? Because it has a conventional shape. You can actually calculate the bounding box in 3D and configure your final acquisition such that it captures the 3D image in the most efficient manner, right? So take-home message from that, sometimes we need more than 2 settings and more than 1 feedback step to actually achieve what we want to really achieve in order to make our protocol optimal. Let's actually now switch from the imaging examples to other type of applications, which we have to do a lot, where we need to do something with our sample on top of just imaging that, right? So, and as I said already, we have a lot of photo manipulation or photo bleaching, photo activation experiments, which I would present, but we also have the projects where we need, for example, to ablate the cells, or where we need, as it was already discussed in the first session, we need to isolate the cells with a laser microdissection and so on and so forth. And again, very often we end up to make more than one decision at the time, right? So you need to define not only the cell, you not only define the position which you move to, but you need to identify the cell to photoactivate. Just to mention a few projects. So one of the projects is for the single cell transcriptomic. And I think this is remarkable because this is also how we combine the expertise of different facilities. And in this case, the imaging is just the first step. What we are interested in this case, we are interested actually to have the cells which have photoactivatable marker. So in this case, it's mCherry. And in this particular project, people were interested to photoactivate cells which has a particular phenotype in a Golgi channel. So green marker corresponds to the Golgi channel. And what they wanted to do, they wanted to separate cells which have more compact Golgi from the cells which have more fragmented Golgi, right? So we were training, we were setting up the feedback microscopy experiment in such a way that we can image the sample, we can identify the right phenotypes, and we can define the photoactivation or photobleaching regions just to photoactivate the right cells. Why we need to do it automatically? Because first, these are live cells, you want to do it as fast as possible. And second, to have the entire workflow done, you need quite a number of cells, obviously, right? So you cannot do just a single cell. So the entire idea of the project, which was done in Rainer's team, is basically to photoactivate large number of cells, then with flow cytometry facility being able to sort them, and finally do the transcriptomic analysis or any kind of follow-up analysis which you want to do with them. So in this case, one can think of it's rather easy. So I need just to photoactivate cells in the given field of view and now needs to go to the next field of view. Let's just look on a little bit more complicated example. We use a photobleaching functionality, but in this case, to do it as FRAP experiment. I'm interested to study the dynamics of the small structures, which are rapidly recovering, doing FRAP. How I would do it manually? Again, classical way to do it on the point scanning confocal microscope, to define your imaging conditions, with which you can catch your desired time resolution, right? To define the region and then starting the bleach experiment. I cannot image the entire cell with the resolution I want at the desired time scale. So what I want to do, I need actually first to identify my region, right, where the structures of interest are located. I need to zoom into that, and I need actually then the second decision, which I have to do manually or automatically, I need to draw this region, and then finally I'm ready to start the experiment after configuring all of that manually, right? And then at the end, if the structure is moving and I still could catch it while bleaching, I can actually track it and record my recovery, and at the end have some kind of a result. The problem to do it manually, again, and that's why the feedback microscopy is important. While I'm configuring all of that, most likely cell will change or structure is gone. It's actually very repetitive, demanding process, but with little degree of success. So again, to do this experiment completely automatically, we actually needed to automate it. But when you automate it, this workflow, which you otherwise would do manually, you require a couple of decisions, right? You need to find the region is number one. You need to define the region, and then you need to trigger that. So at the end, we ended up to do it in a similar way as I showed you in a previous example. So what you see on this diagram, so the top row are the events happening on the microscope side, and the bottom row are the events happening on the analysis side. So we start again with the autofocus, which can be just triggering of the hardware autofocus, but in this case, this was done as yet another feedback microscopy step. So XZ image transferred to the image analysis tool, which identifies the peak intensity for the reflection. Then we have overview image, and then we have image analysis algorithm, which segments our cells, identifies the region of interest. Then microscope focuses in the region of interest, briefly acquires this tag. And in this case, because we are already at the right place with our stage, the communication can be really fast. As soon as we know the target structure, we can, within sub-second range, trigger our bleaching experiment by targeting the right structure and acquire such a data in high throughput manner, right? So at the end, in the overnight experiments, we were able to collect hundreds of such recovery curves. All right, so this is just, again, a summary that we have all these different kinds of applications, right? And again, take home, I want to be flexible with what I decide to select. I want to be fast, but I want to be also expendable on that way. What I want to also highlight, and I want to also discuss later in our discussion session, there are other type of applications which are not strictly the feedback microscopy, but which are coming more and more, is when we want to do something with a sample except doing it with the microscope itself, right? When we want to combine our automated experiment, maybe also the feedback microscopy experiment, with doing certain treatments to our sample, being it either drug treatment, being it multiplexed imaging with labeling with the different antibodies, right? Or very growing and emerging fields doing all this spatially resolved transcriptomics. So what we would need in this case, we would need the external device, which applies a certain treatments, and we would need to synchronize it with the imaging. So not always it requires the feedback microscopy in terms of analyzing the image and finding some target regions and so on and so forth. Although we already have the demand and we already start to get multiple requests, whereas these two sides have to be combined kind of. So you want to synchronize it with identifying the regions. At the same time, you want to be able to synchronize well with the external devices. And just as one example of such work, which was done some years ago with our colleagues from the facility who already left. So in this case, it was a robotics device which was installed on top of the microscope, and sample was sequentially labeled with the different antibodies, with always being in a green channel. So DAPI in this case was, or HERX was used as a reference. But to acquire such type of data, you need fully automated cycle where you have imaging together with the autofocus, right? Then you bleach or wash out the previous stainings, and you have automated staining for the different color or for the different organelle. And this you want to repeat over and over again and to collect such a data in high throughput manner. You can imagine that for certain type of applications like spatial transcriptomics, people want to also target it to a certain regions which would need to be defined automatically for such long experiment. All right, so this was the first part of what I wanted to highlight, basically to provide the range of applications and to provide the range of the needs with what we have to deal with. And then in the second part of my talk, I want to share our experience, right? So what we would need to define such a workflow, right? And which kind of solutions we were actually exploring to enable us to do that. So first of all, in general, so what are the requirements for such a workflow? We would need to have not only the procedure when everything runs, we need also the procedure how to adopt that it's running, right? And how to deal with this afterwards. So first of all, we need like a robust protocols or training how to design and debug this workflow. Sometimes we do it with our users. Sometimes we try to allow users to do it themselves. If we have already similar application which they can evolve. Then we want to have to perform experiments, right? To collect all the data which are needed for the follow-up analysis. And then later on, we want to actually to be able to browse and analyze the data in the consistent manner. So right now, I will focus on the first two blocks because I think the later two blocks will be the topic of the third session. And I would try first to conceptually think what would be the requirements, right? Which I have to ask each particular user or in each particular project, what are the criteria, right, for the tools I would select as the end. So first question, how fast image analysis and communication has to be? Purely will depend on our biology, right? Sometimes running analysis and feeding back to the microscope half a minute will not be a problem. Sometimes you need to have the subsequent range. As I showed you already in multiple of our practical examples, we needed these multiple decisions of feedback steps functionality, right? Not to try to target the specific organelle right from the beginning from the LOSOM image, right? But to be able to make this decision step by step. The project always evolves. We want the possibility to expand the workflows, so we don't want to build them always from scratch. We want to make workflows to certain kinds reusable, right? So if people, instead of selecting one phenotype, select another phenotype, but they want to always bleach, they should not need to rebuild the entire tool, right? So we as a facility are interested to have a reusable workflows and sometimes also transferable to other microscope, maybe of a different brand. We want to log all the experimental info because if I run experiment overnight or over days and I come afterwards and something didn't work, I want to know what didn't work, right? And more information I catch about what Microscop did, but also which decisions were made by image analysis, the better am I to adopt it. And maybe this is also the information which I would need for the analysis, and also how you would manage unexpected events, right? So basically, if something is in delay, what the microscope has to do or what your analysis has to do, right? Not that once your image analysis error appeared, then the entire system stopped for the rest of the night and you come just in the morning and you discover that that experiment actually didn't continue. So all of that had to be managed somehow. So few challenges when, again, talking about particular objects in the facility. So targeting the right objects. Very often people have already their image analysis set up because they use, before they do feedback microscopy, they just image the samples already to classify and to count, right? So very often you are already from the beginning asked to integrate it. So what kind of a feedback you can provide to the microscope, right? Can you only move the stage, or can you also define the regions, or what kind of a control is needed for the particular application? Again, speed of triggering between different modalities as it depends on the application. Synchronization with external devices. So as with the feedback microscope, I feel we are more advanced, but with synchronization with a different kind of devices which will treat our sample, not necessarily devices which are part of your microscope, right? But the devices which you will couple, right, to make the whole workflow automated, for example, with the restaining. And also, which will come probably in the last session, the robust ways to correlate the images acquired with different modalities, probably different microscopes, different brands, and so on and so forth. All right, so now coming closer to what kind of the solutions we can provide for that, right? First of all, and I would really appreciate having all the industry community here. So the amount of the inbuilt solutions growing, and this provides easy access to the novice users, right? And this is also attracting more and more attention to this type of fields, right? Because we have like number of solutions provided by different companies where we have the graphical user interface, and it's easy to design simple workflows. What we find in terms of a facility and in terms of complex projects and growing demand, sometimes we have a lack of flexibility for image analysis. Not always we can support this multi-step protocols where I want to go low zoom image, medium zoom, high zoom image, and so on and so forth. And then, of course, we have to respect development cycle of the company. Of course, if we have a demanding application, we have good collaborations with the companies. You ask, is it possible to do? Yes, but it will be probably coming. But if I have PhD student who needs to finish in a couple of months sitting next to me, so transferring this message to this PhD student probably not the most appreciated task. That's why we have the alternative way. Although from the beginning, I would say we use both ways depending on the complexity of the project and depending on the demand. So the second way, which was already discussed, we can set up the communication between tools, right? We can say we want to have, for example, company software controls the microscope, but we somehow want to dedicate the image analysis to our open source tool or another commercial software. Why not? And making them communicate. So in this case, because you will control the entire workflow with some kind of script, most likely you have the entire flexibility, but most of the time to establish such a workflow, one would need to have the coding required, right? And each time for the particular workflow, we probably would need to code it from scratch. So what we wanted to achieve, right? We wanted to conceptualize this workflow and say, okay, you might need the coding required to do your specific task, for example, for your specific analysis. But maybe if we decide on how the different units will communicate between them, right? We can abstract it in such a way that the requirement of coding will be minimum for each particular project. Right? So before coming to how we would do it in our case, because it's not only we doing that, so it's basically the field which is not, which exists since some time, not since yesterday. I want to a little bit conceptualize, because I was talking to different people, trying to make way similar things. And I think in many cases, I would not say that in all cases, but in many cases, we end up in some kind of architecture, which in one or another way resembles the several modules which we want to deal with. Again, now I talk about the framework of facility when we work with a commercial microscope. So we don't want to control the individual elements of our systems. We want as much as possible the microscope components controlled by the commercial software of the corresponding vendor. So the scheme which we ended up of using for multiple different vendors and multiple microscopes is to have basically the microscope and microscope software provided by the vendor, as we used to have. And normally, if you have the manual workflow, the user will interfere somewhere here, right? Just to control microscope and to acquire the images. So instead of acquiring images manually, we want to acquire the image just automatically. Then newly acquired image can be automatically transferred to the image analysis. In this case, it will be the external software. The external software knows what to do with this image because based on, for example, specific tag in the file name, it knows, oh, it's autofocus image, or it's image to identify the embryos, or it's image to identify the bleaching regions. And then via some command interface, and this has become the critical point here, we'll communicate back to the microscope software what to do next. What is this command interface? So it's typically some kind of a script, which either kind of a macro which runs within the microscope software vendor, right? Or it's a macro which can communicate to normally to another module of the microscope software to provide this type of commands. So why we like to separate these two parts? Our image analysis ideally doesn't want to know what microscope it talks about, right? So our image analysis can actually at this level to define XYZ positions or define the regions, right, which needs to be communicated to the microscope. And basically the task of this command interface will be to translate these commands to the language which is understood by the particular microscope, right? And in this way, we think we can make it modular in order to be workflows truly adaptable. So with last few minutes, I want to talk about how we went about it, but of course, it's only one example how it works. So as I already said, we have a microscope software and we have a remote control interface, which translates our commands, but we want to have very flexible and expandable and customizable workflows on the image analysis side. So for that purpose, I actually over years created a toolbox in Fiji, which allows to do such a custom workflows. So it has a few of predefined workflows, but I see the major strength of it as a toolbox to be with the limited efforts to actually always to adapt it for the new applications by defining the several modules. So when one uses such a toolbox, so one has to pick up from already ready modules or define new with prepared templates. So first of all, the first task which needs to be defined is called job distributor here. So when newly acquired image taken, so this job distributor will not analyze the images. It will just identify this image and basically decide which kind of image analysis has to be done on that, right? Is it just to define the focus plane? Is it just to define the cell and so on and so forth? And then basically the job distributor communicates to other modules or which can be customized as scripts, which will do the particular job. So for standard tasks like finding autofocus, just simple cell segmentation, finding the bleaching row, segmenting the structure, we have already predefined procedures, but the idea is that it will be always expandable. So people can just provide another Fiji script with a certain input and output, or they can trigger any other kind of software, right? Basically by just running the command, running it again via the programming interface. Again, the tool which you will define here will know nothing about your microscope, will know nothing about the feedback microscopy. It will only see the image coming in and provide the feedback in terms of the coordinates coming out. So on top of that, what we want to have, we want to actually collect all this information, right? And in this way, the AutoMicTools takes care of recording the metadata about the experiment. So in this case, I'm not talking about the metadata of the sample preparation and neither of the metadata of image acquisition, which is stored with your image files. It's all the metadata about the decisions. In other ways, coupling your images or image files to the decisions were made in a tabular format, which can be used for browsing and for analyzing the images. And then this is linked just to a simple browser, which can be used in order to be able to browse through the images and see that. So, and this is just example how it will work. So it's tab separated text file, right, which can be also used for the follow-up analysis to know which low resolution image to correspond to which high resolution image, and what were the points used for bleaching, for high resolution imaging, and so on and so forth. As the dedicated regions can be also stored as an imaging ROIs, and in this case, this table will not only know about image files, but also about these ROIs. With a simplistic interactive interface, one can scroll through these images and actually see or control what kind of a decisions Microscop made, whether it was right, whether it was wrong. And especially when you debug the workflow, and if you don't know whether Microscop did the wrong job, or the analysis did the wrong job, right? Or whether both. So it really helps us to understand actually what are the missing points, especially when you tackle the errors which appear not always frequently. All right, so I think at being quite at the end of my time, I would rather skip the rest and jump immediately to the summary. So I showed you in our implementation for such a workflows, which is called AutoMicTools library, which we use for other complex workflows, although we also appreciate have for more simple cases, the inbuilt solutions into the software. And we use it a lot to build the flexible and expandable workflows, right? Especially these are the big projects which are growing over time. We make it compatible with the different commercial vendors, which provide us the interface to communicate actually the commands, right? And we include several of these communication tools into our toolbox. And then by logging all this information, we actually know what a system does. And this is the essential information for me to debug such workflows. The open questions remain. Is it the right way to go? Can certain types of workflows being truly microscope independent, at least on the image analysis and the decision-making side? As I said, and we already heard during the first session, not only we do this, so many people go through the same track, and I would be really interested whether it would make sense to join the effort, right, and to avoid some kind of parallel developments where they can be avoided, especially from the community point of view, right? And I think more emerging topic, which especially with all the spatial omics and all this type of things, synchronizing with external devices like microfluidic devices, roboting pipets, when it's done not on fully customized systems, but when it's done on rather conventional microscope. Can we use the same tool? Can we also use these libraries and have it yet as another milestone, or one would say it's completely different set of applications to also target that? So with that, I would like to acknowledge multiple people. Of course, all our users and collaborators. It's all our inspirations comes from there. I would like to acknowledge the entire team, Rainer, Manuel, and everyone who is here, and few colleagues who were involved before and already left. We work a lot with our Center of Bioimage Analysis to make things modular really helps, because now I can ask, please give me the script which takes this as an input and gives me this an output, and I will integrate this into the feedback microscopy. We have a lot of discussions with our electron microscopy facility, because they basically have the same issues, and this was already mentioned. So electron microscopy would definitely make sense. As I presented, we have multi-facility workflow. So in this respect, we work also with other core facilities. We have a few teams which were involved, like Rainer's team, Corbel Group, and so on, who were involved deeper into developing certain kind of applications. And my special thanks also to Antonio, who is joining us today remotely. So he's now in MPI, but when he was here at EMBL, we had a lot of discussions and a lot of interference with him by doing the feedback microscopy for a certain type of application. So many things I learned from him. So Antonio, special thanks to you. All right, and of course, I would like to acknowledge both industrial partners who make a wish to provide the nice machines and also have these interfaces available. So we have, as I said, we have experience with multiple vendors to do this kind of the workflows, not only in builds, but also by communicating with AutoMicTools. and like Fiji, KNIME, Python, and Elastic communities, where we also get full flexibility for the image analysis which we need. Thanks a lot for your attention, and happy to take questions.